Welcome to a quick review of linkage. Linkage refers to non-random assortment of the allele of genes that are closely located on a chromosome. How do we know that two genes are not linked? Because they assort independently. Take, for example, the A and B genes in a heterozygote, big A, big B, little b. Let's cross this individual to a double recessive, little a, little b, little b. This is called a test cross. We expect 4 genotypic and 4 phenotypic progeny types because the left parent can make 4 types of gametes and the right parent a single type, little a, little b. The no-linkage hypothesis predicts that each genotype should appear with 1/4 frequency. Suppose we genotype 100 progenies. We then use the chi-square test to compare the observed genotypic frequencies to frequencies expected from independent assortment. We observe 22, 26, 30, and 22 instead of the expected 25, 25, 25, and 25. The chi-square for this comparison is 1.76. With 3 degrees of freedom, the corresponding probability of this or more extreme result is 0.62. We cannot reject the null hypothesis of independent assortment, so they are not linked. Now this makes sense. The numbers 22, 26, 30, and 22 do not look that different from 25, 25, 25, and 25. Let's consider the case in which big A and big B are linked at 10 centimorgans, as illustrated. The 2 dominant alleles are on the same chromosome, that is, they are linked in coupling. If they were on different homologs, we would say they are linked in repulsion. In the test cross, we expect the 2 parental genotypes to each have the frequency of 0.45 and the 2 recombinant genotypes to have the frequency of 0.05 each, adding up to 10%. In a real experiment, we get 42, 47, 3, and 8 individuals in each class respectively. The connected chi-square probability is very low, indicating that we can confidently reject independent assortment and that we have linkage, which we calculate as 10 recombinants out of a total of 100 progeny. In conclusion, linkage can be identified by a statistical test and measured as a fraction. There is a constraint. We need sufficiently large numbers, which are easy to get with plants, not so difficult to get with animals. With humans, however, the number of children in a family is not sufficient to evaluate linkage by the chi-square approach. We need a different method.